Okay, let's keep talking about genetics, which is still kind of a big, messy black box at this stage. Brief recap. These are slides that you've seen already, but just as a reminder, Mendel worked out that there's some kind of thingies. He called them elements. We would now call them genes, but they determine what traits pea plants have, whether the pea is tall or dwarf, purple flowered or white flowered, yellow pea seeds or green seeds, whatever the case might be. As we now say, inheritance is particulate. It's a matter of getting these particle-like things that, in our understanding right now, don't change and don't blend. Genes can exist in several forms called alleles. Allele is just defined as different forms of a gene. So we could talk about a pea plant having a gene for flower color that comes in alleles, purple or white. If you want a very simple human example that you could check yourself, although maybe not where other people can see, there is a gene in humans that determines the consistency of your earwax. And there is an allele for wet earwax and an allele for dry, crumbly earwax. So you could very carefully check yourself to see which type of earwax you've got. Although, again, don't do it in public. That's determined by a gene that has two alleles, wet or crumbly. And there's actually quite a lot like that. I think I said earlier that there's about 18,000 human genes. That's an underestimate. The current estimate's over 20,000. That's a lot. Every one of Mendel's pea plants got 2 copies of each of its genes, and it received 1 of each copy from each of its parents. It's the same with you, with one important exception that we will have to wait till Tuesday to get to. You have 2 copies of each of your genes, and you got 1 copy from mom and 1 copy from dad. The 2 copies may be copies of the same allele, in which case we would say that you're homozygous, or they could be copies of different alleles, in which case you would be heterozygous. Alleles may also be either dominant or recessive. And again, dominant doesn't mean better. Dominant just means that if you're heterozygous and you have a dominant and a recessive allele of the same gene, the presence of the dominant allele will hide the existence of the recessive allele. And the convention that we use is we can represent dominant and recessive alleles of the same gene by capital and lowercase forms of the same letter. So you can call the dominant flower color allele capital P, standing for the purple allele of the flower color gene. And you can use lowercase p to represent the recessive flower color allele, which is white in this case. So a white flowered plant must be carrying the allele combination little p. A purple flowered plant could be carrying 2 copies of the dominant purple allele, which we could abbreviate big P, or it could be heterozygous, and we'd write that big P, little p. We use this little writing device called a Punnett square to predict the outcomes of crossbreeding experiments. Sorry, checking the chat one more time. So if we have 2 plants that have genotype big P, little p, they're heterozygous. They're both going to look purple. You won't be able to tell what genes they've got just by looking. You won't be able to see their genotype, as we say. But you will see white flowered plants in the offspring if you crossbreed them. And so we fill in the Punnett square this way. Each parent can pass on one or the other, but not both of each of its genes. So this pea plant up here can pass on either its big P allele or it can pass on its little p allele. It cannot pass on both. So this one will contribute to its offspring either a dominant or a recessive allele. This one will pass on either this big P or this little p, never both. And then to fill in the Punnett square, you put the letters at the top and on the left and fill them in, going, moving them from the top down and from the left to the right. So this square right here represents offspring that got a big P from one parent and a big P from the other parent. This square here represents the ones that got a big P from one parent and a little p from the other parent. This square here represents those offspring that got a little p from one parent and a big P from the other parent. And this square here represents the offspring that got a little p from both parents. And so we predict that 3/4 of the offspring are going to look purple. 1/4 are going to be homozygous dominant. Yes, in this Punnett square, I just got asked, there are 2 heterozygous and 2 homozygous. Yes, half of our offspring represented by the squares here and there are heterozygous. They're going to look purple. 2 of the 4 are homozygous. This square up here is homozygous dominant, and it's also going to look purple. This square here represents the offspring that are homozygous recessive, and they're going to look white. So if you count the number of offspring that you get, you'll get 3 purple flowered pea plants for every 1 white flowered pea plant. You get a 3-to-1. And we can even use these to look at cases where 2 genes at once come into play. So we've got up here at the top and at the left 2 pea plants with the genotype big Y, big S, little s. That's 2 genes. They're both heterozygous at both genes. The Y gene governs the color of the pea, with yellow being dominant to green. And the S gene governs the shape of the pea, with smooth being dominant to wrinkled. Both these plants are heterozygous for both of these genes, and each one will pass on one of each pair of genes, but not more. And there's 4 ways that can happen. The parent up here can pass on the first of each pair, big Y and big S, or it can pass on the outermost of each pair, big Y, little s, or it can pass on the innermost of each pair, little y, big S, or it can pass on the last of each pair, little y, little s. And I use that because at least back when I was learning algebra in the 8th grade from Mr. Ledoux, who was a real jerk, never mind, still scarred though. Back in the Stone Age, when I was learning this, we used that FOIL method to work out how to multiply binomial expressions. I'm assuming that you have something similar in your background. And it's the same for this one over here. This can pass on big Y and big S, or big Y and little s, or little y and big S, or little y and little s. And each P will make eggs and sperm with all 4 of these possible combinations. And if we count them up, what you'll end up with is 9 out of 16 offspring that you get. That's 56.25%, will be plants that produce yellow, smooth peas. 3 out of 16 will be plants that produce yellow but wrinkled peas. 3 out of 16 will be green and smooth peas. And 1 out of 16 will be green and wrinkled peas. Again, we talked about this on Tuesday, but this is just a recap to refresh everybody's memory. You need to know this pretty well because we've got a pretty major lab coming up that's that is going to go a lot more smoothly if you are very familiar with genetics. All right. It's never as simple as it seems. There's always wrinkles. In 1900, a guy named Hugo de Vries and a couple of other scientists, independently of each other, happened to stumble across a dusty old copy of Mendel's scientific paper, realized what he'd done, and started doing experiments that revealed that these basic laws applied to virtually all eukaryotes. I wouldn't be teaching you this if it only applied to peas, but it applies to you. It applies to virtually all eukaryotes. There are some wrinkles, and I'm going to teach them, and I'm going to teach them using cats, because you've all heard of cats, and I'm assuming most of you like cats. I certainly like cats. Preferably medium rare, although they're pretty good in stew. I'm kidding, mostly. We've got 6 of the little bastards at home. But I don't want you to memorize details about how cats inherit things like coat color. You do not need to know that unless you are going to be a professional cat breeder. You don't have to know details of cats. You need to know the general principles that I'm illustrating, because on the exam, I'm not going to ask you questions about cats. I might ask you questions about, corn snakes is a good example. And you'll be able to answer those questions even if you have never seen a corn snake in your life, if you understand the principles that you're supposed to learn from looking at cats. These principles apply all over the place. So don't memorize cat fur color genes. Memorize how you solve, memorize the principles, memorize how you solve problems.
These are the top results.
So here we go. With Mendel's peas, he had the good luck to be working with genes that, as far as he could tell, only came in 2 alleles. So the allele for flower color, or the gene for flower color, is either the purple allele or the white allele. The gene for plant size is either tall plant or dwarf plant, and so on. But there's no reason at all why alleles have to exist only in 2 forms. And if you look at cats, cats can be black or brown or orange or spotted or tabby or things like that. There's lots of possibilities. But if you look at the alleles for dark base color, the gene for dark color comes in 3 alleles, not 2. And we can abbreviate those capital B, lowercase b, and little b prime. The prime is that name of that tick mark next to a letter. And in this case, it turns out black is dominant to medium brown. So big B is the allele for black color. And black is dominant to little b. That's the allele for medium brown. And black is also dominant to little b prime. That's the allele for light brown. Or should I say light brown? And medium brown, little b, is dominant to light brown, little b prime. If cat breeders refer to medium brown as chocolate color and light brown as cinnamon color. I'm perfectly okay with you calling these black, light brown, and dark brown. Or should I say dark brown? All right, one of my cats always says brown when she wants food. All right, so here's what these look like. Up there at the top left, that is a black cat. Below, that's a chocolate cat. That's the medium brown down at the lower left. And a cinnamon brown cat, a light brown cat down there at the lower right. So a black cat could be homozygous and have two copies of the black allele, could have the genotype big B, or the black cat could be heterozygous in one of two ways. A black cat could have a black allele and a medium brown allele, big B, little b, or a black cat could have one copy of the black allele and one copy of the light brown allele, big B, little b prime. So a black cat could have any one of three genotypes. You can't tell just by looking at the cat, although you might get some ideas if you could see that cat's offspring. A dark brown cat could have one of two possibilities, either little b, which is homozygous, or little b prime. And a light brown cat can only have one possible genotype, little b prime. Okay, remember that every cat has only two copies of each gene, one from mommy cat and one from daddy cat. So there could be more alleles than three. There could be 20 or more alleles. It's just that each individual cat is only going to have two at any given, well, not at any given time. For its entire life, each cat is only born with two of the possible alleles. Okay, so each cat itself only has two. What happens when one of these dudes has babies with a white or gray cat? Let me get to that later. That gets into a topic called epistasis, which we will hopefully get to by the end of the day, but might not. A human example, and this is one you're going to hear a lot more about in lab, is the ABO blood group, which was worked out by a guy named Carl Landsteiner, who won the first Nobel Prize in medicine because it made blood transfusions safe. Well, a lot safer. And you may know, especially if you've given blood, that there's four basic human blood types. There's type A, type B, type AB, and type O. Type O is what I've got. And this is governed by having three alleles, which we can just abbreviate with the letters A, B, and O. The A and the B alleles are codominant to each other. If you have an A allele and a B allele, neither one dominates the other. You essentially have both at once, and you have blood type AB. Both A and B are dominant to the recessive allele, which is type O. So if your blood type is type A, you could have genotype AA or genotype AO. If your blood type is B, you could have genotype BB or genotype BO. And if your blood type is O, the only possible genotype for you is OO. And I'm not going to say much more about that, but you're going to work with this in an upcoming lab. That's an example with a great deal of importance for medicine because it determines who you can safely give blood to and who you can safely receive blood from. But again, we will get more of the details in a forthcoming lab. Right. So you can have more than two alleles for any given gene. You can also have something called incomplete dominance. The plants that you're looking at, the flowers are garden flowers known as snapdragons, or in Latin, Antirrhinum majus. And it's probably just as well that Mendel worked with peas and not with snapdragons, because snapdragons might have gotten confusing. If you crossbreed a red snapdragon with a white snapdragon, the offspring are all pink. And this was exactly what I told you did not happen when Mendel crossed his peas, right? When Mendel crossed his pea plants, he crossed a purple and a white, and he got all purples. You know, he didn't get any pink or light purple plants. What he had in that case was a case of complete dominance, where if you have a heterozygous pea plant, the purple allele completely covers the existence of the white allele. That doesn't always happen. Sometimes you will have one allele that is incompletely dominant to the other, and that's what's happened here. Because if you take two pink snapdragons and you cross them, you get a ratio of one red and one white for every two pinks. And the genetics is just the same as crossing two heterozygous purple pea plants. If you cross two snapdragons that both have the genotype big R, little r, some of the offspring will just happen to get the big R from both parents, and they will look red. Some of those offspring will just happen to get the little r from both parents, and they'll be white. And these in the middle are the ones that get a big R from one parent, it doesn't matter which, and a little r from the other parent, and they'll look pink. So instead of that 3-to-1 pattern, you get a 1 to 2-to-1 ratio. So that explains snapdragons. It's a case where the red allele does not completely cover up the white allele, so the heterozygotes are in between the homozygous phenotypes. They're in between being red and being white, which is, well, being pink. If you want a nice kitty cat example, there is an allele in cats for white spotting, and it's in the allele for white spotting, which we can write big S, is incompletely dominant to the allele for solid color, no white spotting, which is little s. So over here on, I mixed up my, no, I didn't either. All three of these cats are basically black cats. They all might well have genotype big B for fur color, because the base fur color is black. This cat on the left, however, is also homozygous for the white spotting allele. So this is a white cat, but the white cat is covered with so many white spots that the cat has basically become a white cat with only a little bit of black. Okay, this is a white spotted cat on the left, right there, this one, where the cat has this homozygous dominant genotype, and the cat has so many white spots that it's mostly solid white. In the center, this cat's going to have the heterozygous genotype, and cats like this have the basic black color, and they will have only a few white spots. Maybe some white on the face, maybe a white star on the chest, maybe just white feet, white socks. Maybe there'll be, we have one of our cats at home is like this. Maybe there'll be a tuxedo cat. Cats that have that white kind of triangle area on the chest are sometimes called tuxedo cats. And then over here on the right, this is a black cat that has the little s genotype, and this black cat is solid black with no spots of white. So there again, the heterozygote in the middle is in between both homozygotes, because that allele for white spotting does not completely cover up the allele for no white spotting. So cats that are heterozygous have only a little white spotting. All right, this is why I sometimes miss not giving these lectures from my house. The internet's a lot more reliable if I do it from school, but I could pick up one of our cats right now and demonstrate that he's almost completely black except for one white spot on his belly, which means that he is heterozygous. And you would all go ooh and ah because he's adorable, and then he would promptly rip my arm open because he's a cat. Okay. Now we get into something called epistasis. And this is very, very widespread. This is a black cat. This cat may well have the genotype big B, for all I know. It could also have the genotype big B, or big B prime. It doesn't matter. We can't tell. But let's say this cat is big B. It's a black cat. However, it's also homozygous recessive for a gene that we can abbreviate with the letter D. And the recessive allele is called dilute. And if a cat is homozygous for the dilute allele, whatever color the cat has is going to get lightened a notch. It actually changes the number and arrangement of pigment granules inside each hair. So a cat that is homozygous for this recessive dilute allele, whatever color that cat is, that color is going to be lightened a few shades. The dilute allele does not determine the color. The dilute allele determines whatever the color already is. And the dilute allele is recessive to the non-dilute allele, which is known as dense and written as capital D. That's epistasis. There's no gene for gray fur color in cats. There is a gene for black fur color and a gene for make the color lighter. And that's how you get a gray cat. And that's epistasis, several genes acting together to affect the same trait. And that's how you get a gray cat. So for example, this cat is on the left is a black cat that is also homozygous for the dilute allele. This cat right here on the right is a medium brown cat who frankly looks really PO'd. But that's a medium brown cat who also is homozygous for the dilute allele. So this cat is no longer medium brown. It's a different shade of light brown with these copper colored eyes. And cat breeders refer to this as lilac for whatever reason. I don't know why they call it that. Okay, so there are no genes for gray fur or lilac fur. There's genes for black and brown, and there's a gene with an allele that will lighten the basic color a couple of notches. That's epistasis. By the way, if anybody was wondering about this, humans do not really come in a narrow range of colors. We are variable. You know, these kids range from very pale over on the left to very dark over on the right. And, you know, there's a lot of in-betweens right here. The reason for this is last I checked, there are 9 different genes that all have an effect on human skin color, and each of those genes has 2 or more alleles. So to work out the skin color for crossing 2 different humans, you would need I think it's a 2, 4, 18, you need a 1024 by 1024 Punnett square, right? There's a lot of possible phenotypes, and this is why human skin color doesn't really fall into neat classes. We're used to thinking of people as being black or white, But the truth is, both the people that we call white and the people that we identify as black have quite a bit of range. Some of those alleles tend to be more common in people who came from Africa, and other alleles tend to be more common in people from Northern Europe and so on. But there's no place where you can truly draw a dividing line. Human skin color is a very epistatic trait, meaning there are at least 9 genes that affect it. We're a lot more complicated in this regard than peas. Pea flowers can be either purple or white, but really we're not all either white or black. We're not, apologies to Paul McCartney, we're not really ebony and ivory. Quite a lot of people fall in between, and that's just fine. That's just the way it goes. Because when you're talking about genetics, it don't matter if you're black or white. Ow! Sorry. Okay. I had to do the Michael Jackson ref. So that's an example of epistasis. 2 or more, sometimes a lot more genes governing a phenotype. The same applies to things like your height, your build, possibly aspects of intelligence, although people argue about that a lot. There are genetic effects on those, but there's no single gene that will make you a basketball center or a Kentucky Derby jockey. There is no single gene with alleles for being tall or being short like there are in peas. There's a bunch of genes, and they interact in ways that are not easy to disentangle. So height tends to run in families. Tall people on average will have tall kids, and short people on average will have short kids. But there is no single allele that determines how tall you're going to be. The same is true for your build, aspects of intelligence, although there's a lot of environment involved in that too. Quite a lot of things are genetic, but they don't break down to nice, neat Mendel's P type examples. A second type of wrinkle you can get is called pleiotropy, and that refers to cases where one gene has several effects. Epistasis is several genes contributing to one trait in the phenotype. Pleiotropy is one gene having several effects on the phenotype. And this is a cute example from cats. You're looking at a Devon Rex cat. Rex cats have wavy fur. If you were to pet this cat, it's hard to see in a picture like this, but if you were to pet this cat, you'd feel the difference right away. Most cats have straight fur. Devon Rex cats are wavy. And this happens to be a trait that is governed by a single gene with 2 alleles. The Devon Rex allele is recessive to the not Devon Rex allele for straight fur. So we write the Devon Rex allele as R with a little E superscript. And the non-Devon Rex allele is big R with a little D superscript. So you could predict what happens if you crossed a Devon Rex cat and a non-Devon Rex cat. This is pleiotropy because of this. Here's a bunch of Devon Rex kittens, and they either look absolutely adorable or kind of strange, depending on your point of view, because if you're a cat and you're homozygous for Devon Rex, you get wavy fur, but you also have this odd shape of your skull where your ears are huge and set low on your face, and your face is unusually wide. These are sometimes called fairy cats or pixie cats or, Yoda cats. These cats look like the Mandalorian is trying to get them through a war zone. I'm imagining all of you going, "Aww," right about now. And all of those effects are due to just one allele. If you are homozygous for the Devon Rex allele, it affects not just fur texture, but it also affects skull shape and ear shape. One gene, several effects. Somewhat more poignant example of pleiotropy, the question I was asked earlier about what happens if you cross a black cat with a white cat actually is tricky because there are several different ways you could get a white cat. Not all white cats have the same genotype. But there is a completely separate allele or a completely separate gene with two alleles, which we can write big W and little w for white fur. Cats that are homozygous for the recessive white fur allele are completely white. They also have blue eyes, and they're also usually deaf. So that's pleiotropy as well. It affects not just the fur color, it affects development of key parts of the middle ear. Cats with blue eyes and white fur often are born deaf, which is just one of those things. And by the way, if you were to cross a black cat and a white cat like this, you would get white offspring because the white allele interacts epistatically with pigment and basically cuts off the cat's ability to make any sort of pigment at all. But that doesn't mean white is dominant to black, because black and white are two different genes. It means that white has epistatic effects on black. But the answer to that question is even trickier because, again, there's more than one genotype that will give you a white phenotype. We have two cats at our house that are white all over, but they have green eyes and they're not deaf. I mean, they ignore me a lot, but all I have to do is open a can of cat food and they've got very sensitive hearing. Little bastards. Anyway, sorry. So yeah, the answer to I can't give a simple answer to the question that was asked. But this is one type of pleiotropy where one gene affects not just fur color, but ear development. And I'll go ahead and give you a dog example for the dog people in here. Many breeds of dog can show a coat pattern called merle. For some reason, in dachshunds, it's called dapple, but it's the same underlying trait as merle. And merle is this. It's a coat pattern in which large patches of the base color are lightened. So this is a mostly black dog, except he's also got the merle phenotype, and you can see much of the area on his fur that would be black has been lightened to gray, and you have these patches of black in the middle of the gray. That's pretty typical for some breeds of dog. You'll see this pretty frequently. And merle is dominant to non-merle. The problem is that if you have a dog that is homozygous for the merle allele, genotype big M, that dog not only lacks pigment, but usually that dog is going to be blind and or deaf. The alleles for pigment development in dogs and in cats also have an effect on the development of the inner ears and sometimes on the eyes as well. That's pleiotropy for you. One gene can affect multiple aspects of the phenotype. And for this reason, it's considered unethical to crossbreed two merle dogs because two healthy merle dogs, like this one, are going to have the genotype big M, little m. They'll be heterozygous. And if you cross two merles, 25% of the offspring will just happen to get a merle allele from mom and a merle allele from dad. They will be double merles, and they will potentially look like this. They'll be born with abnormal eyes and abnormal inner ears. And it's kind of unethical to crossbreed dogs if you know that a sizable fraction of the puppies are not going to lead normal lives. So if you're ever breeding dogs, try never to crossbreed two merles. There's an even more extreme example from cats. This is a Manx cat, and Manxes are born without tails. There are other breeds of cats that are born without tails. There's a breed called the Japanese bobtail, for instance, and this does not apply to them, but it does apply to Manx cats. The tailless allele, which we can write as capital T, is dominant to the tailed allele, which we can write as lowercase t. Okay, so tailless is dominant to tailed. But if you crossbreed two Manx cats, you end up not with 3 tailless cats for every tailed cat. You get 2 tailless kittens for every kitten with a tail. And the reason is that the big T genotype is an embryonic lethal. It messes up the development of that embryonic cat inside mommy so much that it miscarries. Embryos with the genotype big T are never born. They don't develop to full, they don't develop to maturity. I'm not sure if they get miscarried or some species can absorb fetuses into the body. Humans can't, but some mammals can. I don't remember if cats can. Regardless, you will never see a cat with the genotype big T. They never develop past an embryonic stage. So a tailless cat, a Manx cat, has to have the genotype big T, little t. So if you cross 2 big T, little t cats, one of the offspring, or 1/4 of the offspring, will have the genotype big T, but you will never see those because they never get born. That leaves 2 out of 4 offspring will have the genotype big T, little t, and they will be Manx cats. And 1 out of the 4 will have the genotype little t, and those will be Manx cats, except they will have tails. So that's one more thing that can distort results, is if you have alleles that are embryonic lethals. They will give you, it's a good thing Mendel did not see any examples of this. There are some examples in plants, but they could have thrown him pretty astray if he'd had the bad luck to be working on a trait in peas that had embryonic lethals. Okay, last thing, I promise. Well, last thing for now. There's an allele in cats that's just called the color allele. And the color allele, if it's present, allows all of these other color genes to be expressed. So if a cat has at least 1 copy of the dominant big C allele, then the cat can show colors like black or brown or orange, which I will actually have to talk about later, or gray or lilac or calico or tortoiseshell, which I will also talk about later. Whatever color the cat is, the cat will be able to show it. That color allele is dominant to an allele called albino, which we can write as little c. And if a cat is homozygous for the albino allele, all possibility of making color is cut off. That's going to be a white cat with pink eyes, which you don't often see among cats, although you get the same gene in mice, for example. White mice with pink eyes are not that uncommon. And that's yet another example of epistasis. There has to be 1 copy of that color allele to allow any color to be expressed in a cat. And that's genes working together to determine the phenotype. However, there's another allele of this gene that's often written big little c with a superscript s, little cs right there. And if a cat is homozygous for the little cs allele, then whatever color that cat has will be expressed, but only on the parts of the cat that are cool. That little cs basically puts a temperature triggered switch on the cat's skin cells and essentially says, if the temperature is warm, don't put any color in the skin or in the fur. If the temperature is just a little bit cold, then you can start putting color there. Now think about what this looks like. The parts of a cat that are cool are the same as the parts of you that get cold. So if you're walking around and a cold front comes through and suddenly it gets chilly, what are your body parts that are going to feel it first? Well, probably your feet, probably your fingers, and probably your face, right? Your nose and your ears. And what you would get in a cat is the Siamese pattern. On the left, that's a cat that is homozygous for the CS allele. It's basically a black cat, but the only places where you see the black color are the cold spots, the face, the ears, and you can't see it in this photo, but you'd see black on the feet, black on the tail, and black on the testicles if it's a tomcat who still has them. Over there on the right, that is a light brown cat. But that cat is also showing its light brown color only on the face and the ears, and off camera also on the feet and the tail and the scrotum if the cat's still got it. Pigment is only showing up on the cold parts of the cat's bodies. You could take a Siamese cat and shave the fur off of a patch of that cat's back and put a cold pack on that shaved patch of skin and keep it there for a while. And you would end up with an extraordinarily pissed off cat that would crap in your shoes. But you would also end up with a cat where the fur would grow in dark. If you artificially chill part of a Siamese cat's skin, the fur will grow in darker. In fact, I have never seen this myself, but when my wife was a girl, they had a Siamese cat at the house, and she was an outdoor cat. And in winter, that cat would turn noticeably darker. And then the cat would lighten up in summer, because in winter, the cat was just being exposed to colder temperatures. And all of the cat would turn darker. And again, I've not seen this personally, but she tells me that's what's going on. Pigment will only appear on the cool parts of the cat's bodies. So that's a case where the environment that the cat is in has a direct effect on the phenotype. The temperature triggers the cat to develop pigment in cases where the cat has a particular gene. This is actually a case of, this is a phenomenon we call phenotypic plasticity. Don't memorize that term. But there's lots of cases where phenotype is not determined only by the genes and not only by the environment, but by both of them together. Bet you never knew that Siamese cats were such complicated beasts. And by the way, you can have the genotype little cs, and it doesn't matter what the other color genes are. We had this stray cat that wandered into our house in February that has adopted us. I swear, my family did not set out to get 6 cats. 6 cats have happened to us. And the latest one to happen to us is a tabby cat with stripes, and she is a mix of dark and orange, and she is a Siamese. So she is basically a heterozygous genetic experiment. And that's putting it mildly. But she also shows the darkest colors and the striping of her tabby patterns almost entirely on her face, her ears, her tail, and her feet. And the rest of her is a much lighter kind of cream color pattern. And by the way, if you're sick of cats, here's a non-cat example. I took this picture in one of the Kroger supermarkets in town a couple of years ago. They were selling potted hydrangeas in the spring. These are hydrangeas, pretty popular around here. You'll see them in people's gardens. They grow pretty well in our climate. And we have hydrangeas that are purple and pink. And hydrangea plants can only make blue pigment when the soil is a little bit acid. Not super acid, you know, not like lemon juice or anything like that, but only when the soil is a little bit acidic does that free up the minerals that hydrangeas need to absorb in order to make blue pigment. If you grow your hydrangeas in more alkaline soil, less acid, they will make pink flowers. And the color difference is not genetically determined. You can make your hydrangeas change color if you alter the soil. You can turn your pink hydrangeas blue by putting iron in the soil. And that has the effect of absorbing some of the alkalinity and making the soil just a little bit more acid. So mess with the color of your mess with your soil chemistry, and you can change the hydrangea color. The technical name for this, by the way, is phenotypic plasticity. By the way, the white hydrangeas are genetically different. They don't produce any pigment at all, no matter how you treat their soil. But the colorful hydrangeas can be either purple or pink or kind of an in-between, depending on the soil chemistry. So yeah, nice example of this that you could see in any garden store in spring, or even in the supermarket in spring. And if you think about it, human skin color is kind of like that as well, because it's influenced by a bunch of genes, but it's also influenced by the environment, namely how much time you've been spending in the sun or hitting the tanning bed, right? So the exact tone that your skin takes depends largely on genes, but also on how sunny your environment is and how much sun your skin is being exposed to. So that would be yet another case of phenotypic plasticity. In lab, you'll see the effects of single genes with simple effects. Mendel's pea plants are fairly simple. But the truth is that practically all genes in the real world show some degree of epistasis and some degree of pleiotropy and some degree of influence by the environment. And the reason this is important for you guys to know is there is still sometimes a lot of hype about scientists discovering or trying to discover the gene for mental illness, the gene for depression, the gene for breast cancer, the gene for obesity, the gene for Alzheimer's, things like that. I'm not saying that's not important work, but there's actually very few human genes or human disorders that we can completely pin down to the effect of a single gene with a couple of alleles. Most of our traits are not nearly as simple as the ones that Mendel demonstrated. That doesn't mean Mendel is wrong. it means that Mendel's work was incomplete, and we have a fuller understanding of how genetics works. So if you hear claims that scientists have found the gene for dandruff or whatever it is that they're working on, that usually is bad science reporting. Usually that has more to do with the press wanting a story than it does with what the scientists are actually trying to do. So that beanbag genetics model where each gene is responsible for exactly one trait is a simplification. It can be useful at first, but the real world and real organisms are more complicated than that. All right. Looks like I've still got you folks for 14 minutes, so we are going to march boldly onwards, except not this Siamese cat. This cat does not want to march boldly onwards. This cat is going to take a nap, and let's move on. So we've been shaking this black box, and we've established that there's these things called genes, and the question rises, what are those genes? What is inside this black box that we keep rattling around? We have to go back in time to the 1600s, to the work of a gentleman in London named Robert Hooke. Hooke is known as a physicist. He made discoveries in fields like elasticity, in developing calculus. He designed buildings and he composed music. He was gifted in a lot of different areas. And he was trying out a new invention of his called the compound microscope. Some of you have already gotten to use the modern compound microscopes that we have in lab. Others of you will get to either this week or next week. And you may have noticed that the modern compound microscope is not quite as sophisticated as Hooke's. Although they work in fundamentally the same way. You have multiple magnifying lenses, all in one light path. And Hooke was the very first person to see and name cells. These are drawings he made that he published in 1665, drawings of a thin slice of cork, the stuff you put in wine bottles, of course. But cork is the inner bark of a species of oak tree called Quercus fellis, called, you will be surprised to hear, the cork bark oak. Duh. So cork is a plant tissue, and Hooke could see that this plant tissue, looked at up close, is divided into many little chambers that reminded him of the cells in a monastery, or we might now say the cells in a prison. So he called these little openings inside cork cells. Turns out that not just plants are made up of cells, but animals and fungi are as well. Animals, of course, and also plants are made up of differentiated cells. Your oak tree might be made of about 50 different cell types. You're made of roughly 200. And human sperm cells were first discovered in 1677 by a Dutch gent named Antony van Leeuwenhoek. For reasons that I trust I don't have to explain, it took a lot longer to find egg cells. Those were not seen until the first ones were seen in the year 1797. Egg cells constantly stay inside the female body, whereas it's pretty easy to get sperm cells to leave for examination. One early idea that people had was that either the egg cell or the sperm cell, people debated as to which, contained a tiny duplicate of the parent. So what you're looking at is what somebody thought he saw in a sperm cell back in the 1600s, and thought that the head of the sperm cell contained a tiny little person and the sperm cell would, bury itself in the wall of a woman's uterus, and that tiny little baby inside the sperm would inflate and get bigger and bigger and bigger, and eventually become a baby and get born. And then if this baby's a male, then this baby already has testicles that has the next generation inside. And then each of those contains the next generation inside its testicles. And then each of those contain the next generation inside its. So humanity is basically nested one generation inside the other, like Russian dolls. Now, this is a pretty serious matter, because if this is true, this means that every healthy teenage boy kills about 20 million babies every day, and sometimes twice a day. But the development of better microscopes basically showed that this hypothesis is false. People do not come from teeny, tiny little babies scrunched into daddy's sperm cells. It does not work that way. Okay, so how does heredity work? We'll skip over a couple of centuries and jump to 1839, when a couple of German guys named Schwann and Schleiden proposed that all living things were made up of these living subunits called cells. There are some living things that are made up of only one cell, many protists, for example. There are some living things that are made up of a great many. You're made of, what is it? I think it's 37 trillion. So the number of cells may be one, or it could be enormous, but they're still cells. They originally stated that a cell was defined by having a nucleus. Here's some diagrams that they drew. We now know this is not true for prokaryotes. It certainly is true for eukaryotes. And as Schleiden put it, looking at plants, although the variety in the external structure of plants is great, their internal structure is simple. This extraordinary range in form is due only to a variation in the fitting together of elementary structures, which indeed are subject to modification, but are essentially identical. That is, they are cells. So plants range from mosses to flowers to oaks to redwoods. They have a huge range of size and shape, and yet the cells that they're made of are not really different from each other. The difference is just in how you arrange them. Compare this to the UCA campus. We have small buildings and we have big buildings. We have buildings for teaching. We have buildings for recreation. We have the student center. We have buildings for administration. We have small buildings and we have big buildings. And yet they're all basically made of the same parts list. They're all made of 2x4s and brick and the same types of wiring and things like that. You can build a ridiculously large number of possible buildings out of a relatively small number of different components. Schleiden is pointing out that the same thing applies to plants. There's not that much difference in the cells of a little moss from the cells of a giant oak tree. The main difference is how you put them together. Well, there is the difference that mosses don't have vascular tissue. So let's say there's not much difference between the cells of a single grass and the cells of a giant oak tree. The difference is in how you fit them together. Okay. They originally thought that cells might come into existence by some sort of crystallization process. But a guy named Virchow worked out and stated in 1855 that cells could only come from other cells. Cells don't spontaneously appear. at least not under any conditions that we have on Earth now. As he put it, omnis cellula e cellula, all cells from cells. Jump to the 1870s, and microscopes have gotten a lot better. That 1840s microscope is not very good. That 1880s microscope is about as good as anything we can make now. Microscopes by this time are starting to run up against the physical limits of what you can do with light. And this was making it easier to see structures inside cells. The other thing that's making it easy is in 1856, this British guy named Perkin invented a chemical that he extracted from the waste products of the coal industry. And he was trying to make medicine, and it failed, but the failed color turned out to be this brilliant purple. He named it mauve, M-A-U-V-E, by the way. That was the first of the artificial dyes to be invented, although lots of people followed up and found more once Perkin had shown them where to look. So by the 1870s, there's a big artificial dye industry. This is where we get, we're all used to being able to go out and buy clothes inexpensively in any color we want. This only became possible with the invention of aniline purple dye. Before that, dyes were expensive and they were made from plants, and there was only a limited color selection available. The reason this matters, not just for the history of fashion, it's some artificial dyes turned out to be useful in looking at cells under the microscope, and some dyes would stain certain colors, but they would not stain others. So you could pick out structures in a cell if you stained it. Many cells are pretty much transparent under the microscope, but if you add stain, you can bring out the colors and you can see things that you couldn't.
Ethan, right?
What?
We have
Okay.
class today in history?
What?
We have class today in history?
Mute your mic.
Where?
History class.
Somebody
history
mute your
with
mic.
Miss
Somebody's talking.
Little?
Yeah.
Please mute.
Yeah, we do.
All right.
Okay, please mute. All right, I'm almost done, I promise. 1873, a guy named Schneider is looking at cells in the developing tail of a salamander. And he notices that if you look at the nuclei, there is something weird. Ordinarily, the nucleus, which is right here, just kind of looks like a round blob. But cells have to come from other cells. So one cell, to make more, has to divide in two. When a cell divides, the nucleus seems to turn from being a blob into a set of stringy objects, which move in some kind of pattern and then turn back into a round, grainy blob. So here are those stringy objects there in the middle. They line up in the middle. And then here's those stringy objects moving to opposite sides of the cell. And then here, once division is done, here we have two clumps of stringy objects right there that are going to form two new nuclei. Now, we can't get away with calling these things stringy objects, but it turns out these stringy objects tended to be very strongly colored by many of these stains, these artificial dyes that had been invented. And because they tended to take up color very strongly, whereas other parts of the cell usually didn't, they came to be called colored bodies, or in Greek, chromosomes. And we will end there and start talking about chromosomes next Tuesday. All right, we'll hereby stop the recording.